How are genes transferred between organisms? That is a question that we aim to address with this lecture. Let's start by taking a look at how humans do it. So here's a picture of me, and here's a picture of my husband. I have poor hearing, I am hard of hearing, and that is because of genetic defects in the way that my cochlea are structured. My husband has excellent hearing. His cochlea are structured correctly to pick up even small sound waves, and therefore he can hear a lot better than I can. So if I had different genetics, let's say I had his genetics for the cochlea in the inner ear, I should be able to hear just as well as he does. But I would need those genes. I would need those alleles more specifically, and I don't have them. Wouldn't it be great if I could simply go up to him and ask him to hand over some of that DNA that I could somehow incorporate into the cells of my cochlea so that they express these new stereocilia? That would be cool, but it's not something that we're capable of doing. In other words, humans are not able to exchange DNA in a way that modifies their own genetics. However, there is obviously some gene transfer, or we wouldn't have the diversity that we see. So although we are unable to modify our own individual genetics, we can create new genetically distinct individuals through sexual reproduction. I can give my DNA, and he can give his DNA to a new individual. And there's a picture of my son when he was 2 years old. And we got lucky. He also has excellent hearing. Sexual reproduction leads to what is called vertical gene transfer. And what that means is that in order for genes to be transferred, you need to go down to the next generation. Again, what that means is individuals, one person here and one person here, cannot exchange DNA back and forth. Rather, you have to wait and create a new genetically distinct individual through sexual reproduction to transfer your genes. Prokaryotes don't do this. Prokaryotes do not do sexual reproduction. So what do I mean by sexual reproduction? If I take two bacteria and put them together, they can't create a new baby bacteria that has a mixture of each parental bacteria's genes. That's not a possibility. So bacteria do not do vertical gene transfer. However, they do have the ability to do horizontal gene transfer. Horizontal gene transfer is where an individual bacterium somehow acquires new DNA or new genes and incorporates it into their genome. Kind of like the example I started with. If I could do horizontal gene transfer, I should be able to pick up new DNA that codes for quality cochlea in my inner ear, and incorporate that into my genome so that my cochlea are improved and I can hear better. Bacteria actually can do this, and it is called lateral or horizontal gene transfer. So let's go ahead and define these. So the important thing to understand is that in horizontal gene transfer, the individual or the cell that acquires this new DNA is genetically modified. So they've now modified their own genetics. And anytime you have modified genetics within an individual or within a piece of DNA, we call that a genetic recombinant. So a genetic recombinant is going to be an organism, a cell, or even a piece of DNA that is a combination of the original DNA plus new DNA from a different organism. So the term recombinant means to recombine, to combine two different sources of DNA into a single piece or a single genome. So bacteria are able to do this, and they have three different techniques by which they are able to do this. We give them names. Here are the three different ways in which bacteria can achieve horizontal gene transfer to become genetic recombinants. We have transformation, conjugation, and transduction. Transformation and conjugation are going to get their own lectures. Transduction is perhaps the most complicated of the three and requires some knowledge about how viruses work, so we're going to save transduction for when we get into the viral section of this class. But briefly, I will say that it is an accidental injection of new DNA from a virus. In other words, a virus brings some novel DNA and injects it into an unsuspecting bacterium. Transformation and conjugation will be covered in lectures 6D and 6E, respectively. Now, vertical gene transfer is going to be creating a new individual. So the only way that genes are transferred is by creating the next generation. And on the family tree, that means going down, and that would be vertical. Okay, since our focus is prokaryotes, let's come back down to this genetic recombination idea and think about some ways in which this might occur. So imagine if I have a bacterial cell and they have a chromosome, and that's all their original DNA. There are essentially two major ways in which that cell can become a genetic recombinant. Sometimes we just use the term recombinant for short, and we assume the word genetic is implied. So one way is to basically take a separate piece of DNA carrying a small number of genes. These separate extra chromosomal pieces of DNA are known as plasmids. And in lab, we learned that bacteria have two ways to acquire plasmids: transformation, which is covered in lab 16, and conjugation, which is covered in lab 14. So this bacterium is now a genetic recombinant because it has its original DNA in blue and new DNA in pink. But it turns out that that's not the only way that bacterium can become a genetic recombinant. Another way is if it gets some new DNA that is chromosomal. Now, it's not going to get a separate chromosome, but it might actually get a piece of a chromosome, and it can swap out its old DNA for the new DNA at those particular genes. So let's say that instead I have a single piece of DNA here that is really part of the chromosome. It's not a plasmid, it's not self-contained. And through one of the mechanisms that I described above, this piece of DNA gets into this new cell. It's not a recombinant yet because, for reasons that are really beyond the scope of this class, if that DNA doesn't find a home, it's eventually going to be cut up by things called restriction enzymes. These are enzymes that the bacteria has that cut up that DNA, and it will eventually be degraded. So it wouldn't be a recombinant unless that piece of DNA was able to somehow get into the chromosome. So this is a little bit harder to draw. So if we have a piece of DNA, then if that DNA matches something kind of similar, like it might kind of run around and look for something similar. Let's say it matches pretty closely here. In other words, it carries a pretty similar set of genes. then we can actually eliminate one of those strands and fuse this DNA in its place. And now that that new DNA has a home, we would say that this cell is in fact a genetic recombinant. Another look at this idea of swapping out pieces of chromosome is shown here. Here I have one individual and a second individual's DNA. And you don't need to memorize these steps, but what I want to show you is that some of this DNA is going to basically bind to that DNA and they're going to swap. So both of these are now genetic recombinant. Again, for reasons that are beyond the scope of this lecture, this really only happened when the DNA is pretty similar. So this is a very brief introduction into the concepts of genetic recombination. The major takeaways here are that bacteria do horizontal gene transfer, or also known as lateral gene transfer. Eukaryotes are only capable of doing vertical gene transfer. And since our focus is generally bacteria, we're going to be looking at the prokaryotes. There are 3 mechanisms of horizontal gene transfer. And if horizontal gene transfer is successful, you create a genetic recombinant. The genetic recombinant can be created by a cell either taking in a new plasmid, which is kind of a self-contained piece of DNA, or by swapping out part of their old chromosome for a new chromosomal piece from a different organism. That concludes this lecture. Thanks.